Good afternoon, everyone. Welcome and thank you for standing by. All participants are in a listen-only mode until the question and answer section of today's webinar. Today's call is being recorded. If you have any objections, please disconnect at this time. I am Nikki Grimsley with the Clinician Outreach and Communication Activity, or COCA, with the Division of Emergency Operations at the Centers for Disease Control and Prevention. We are delighted to welcome you to today's webinar. What has Toxoplasma in sea otters taught us about risks and the One Health approach to global health? COCA is excited to offer this call in partnership with the American College of Veterinary Preventive Medicine. We are pleased to have with us today Dr. Heather Fritz. Free continuing education is offered for this webinar. Instructions on how to earn continuing education will be provided at the end of the call. At the conclusion of today's session, we will be able to describe the life cycle of Toxoplasma gondii and the importance of oocyst in transmission, explain the different mechanisms for oocyst accumulation in the ocean where sea otters become infected, list possible steps to reduce pathogen pollution in coastal habitats, and define a keystone species and discuss what we have learned about ecosystem health, and human health risks from studying sea otter health. CDC, our planners and their spouses and partners wish to disclose they have no financial interests or other relationships with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters. Planners have reviewed content to ensure there is no bias. At the end of the presentation, you will have the opportunity to ask the presenter questions. You may submit questions through the webinar system at any time during the presentation by selecting the Questions tab on the webinar screen and typing in your question. Or you may click on the Raise Your Hand icon on the webinar screen and your line will be unmuted so that you may ask a question. Our presenter today is Dr. Heather Fritz. Her dissertation work focused on the molecular characterization of the Toxoplasma gondii oocyst. After completion of her PhD in 2011, she continued research projects directed at developing reagents and methods to concentrate and detect Toxoplasma oocyst in environmental samples and molecular investigations of the biology of the oocyst stage to study oocyst environmental resistance, as well as mechanisms of host cell invasion during oocyst sporozoite-initiated infections. For more than 10 years, she has worked with a team of experts in the field to better understand how Toxoplasma oocysts accumulate in the environment and infect sea otters. At this time, we welcome Dr. Fritz. Dr. Fritz, please go ahead.
Okay, thank you. I hope everybody can hear me well. It is a privilege to be able to participate in this webinar. I do want to upfront just acknowledge that much of what I will cover in this talk is really a product of research that was done by many different collaborators, both here at UC Davis and at other institutions. So with that, I'll first start with a description of what we mean when we talk about global health. And there are a lot of different definitions that you might come across. I'm just going to use the one that's available on Wikipedia. And Wikipedia describes it as the area of study, research, and practice that places a priority on improving health and achieving equity in health for all people worldwide. It is meant to address problems that transcend national borders or have global political and economic impact. So global health is about worldwide health improvement, reduction of disparities, and protection against global threats that disregard national borders. And it's not meant to be confused with international health, which is defined more as a branch of public health that focuses on developing nations and foreign aid efforts by industrialized countries. Okay, so I'll also refer to One Health, and what One Health is defined as is an interdisciplinary approach to solving specific complex problems that arise at the interface of animals, humans, and the environment. So here is a depiction of what that may look like, and it's really examining the overlap between animal health, human health, and then how they intersect with global ecosystem health. So it's meant to be holistic. By nature, it's collaborative, and it's very solution focused. So where does that bring us next with the story of the southern sea otter? So more than this being a very charismatic and popular animal in coastal California, the sea otter has actually taught us a lot about One Health and how a One Health approach can help us answer very complex questions. So to give you a little bit of background on the southern sea otter, it was federally listed as a threatened species in California, and it's found only along a small region of the central coast of California. The total population is about 3,000 animals. Sea otters are considered a keystone species. In ecology, a keystone species is one that has a disproportionately large effect on the communities in which it occurs. Those species help to maintain local biodiversity within a community, either by controlling populations of other species that would otherwise dominate the community, or by providing critical resources for a wide range of species. Other species in the biological community depend on the presence of keystone species to maintain the community structure. And the community structure in which the sea otter resides is one that is inhabited by a variety of other marine mammals, fish, birds, marine plant life, and sea urchins shown here, and of course is used heavily by humans. So the importance of sea otters relates heavily to kelp forests, and kelp forests are important in the coastal ecosystem because they protect the coast from erosion and they provide habitat for a wide variety of animals. And where the sea urchin comes in is it preys upon kelp, and sea urchins will destroy kelp forests quite rapidly in the absence of sea otters, which prey on sea urchins. So that is one way in which the sea otter helps to maintain the integrity of that ecosystem, is by keeping the sea urchin population in check. So I thought I would give you a little background on how Toxoplasma gondii relates to this story of the sea otter. And I'll say personally, I became interested in Toxoplasma gondii oocysts when I was doing my PhD work, because the impact that Toxoplasma infections were having on the recovery of the southern sea otter population in California had only recently been discovered. So sea otters were hunted pretty heavily for fur, and their population was nearly wiped out until around this point here on the graph, early 1900s, when they were listed as a protected species, a threatened species, at which point their population began to recover. However, their population failed to recover at what would be predicted rates. So the carrying capacity is estimated more at about 16,000 sea otters, and yet their population is kind of hung out down here in the lower range of about 3,000. So researchers took a closer look at why sea otters were either failing to recover in population or what was killing sea otters. And what they found was that infectious diseases were among the most important causes of sea otter death, and among those, protozoal parasites were important. And Toxoplasma gondii is one of the two protozoal parasites that are known to cause severe disease and death in sea otters. And a study that was done in 2002 found that about 38% of live sea otters were seropositive for Toxoplasma, meaning that they had been exposed and probably had chronic infections. And about 52% of the dead otters were seropositive for Toxoplasma. So the question was, how is a terrestrial pathogen causing such profound disease in marine mammals? And this story gained a lot of press around the time when it first became known. And this is just one of the articles that came out, and it resulted in quite a lot of response from the public about how to manage feral cats, and a real interest in determining how oocysts were actually reaching the ocean to infect sea otters. So you're probably familiar with Toxoplasma, but the other parasite that is causing severe disease in sea otters is Sarcocystis neurona. Interestingly, for both of these parasites, they have only one definitive host. A definitive host is the host in which sexual reproduction occurs, resulting in excretion of the environmental form. So cats excrete Toxoplasma oocysts in their feces, and opossums are the only host for Sarcocystis neurona, which is a closely related protozoal parasite. And somehow these oocysts are making it from land to sea. A little background on Toxoplasma. This is a really fascinating organism, I think. It was only discovered a little over 100 years ago. It was first discovered in the North African Gundi rodent. It was given the name toxo for bow and then plasma form because of its shape. And this is a tachyzoid, another one of the life stages for Toxoplasma. As you know, it's a protozoan parasite, belongs to the phylum Apicomplexa, and it's an obligate intracellular parasite. And once it's infected a host and lives within the cells, it interacts with host cells via secreted proteins that are in the apical complex up here in the parasite, and that is also where classification of Apicomplexa comes from. So the secretory organelles of Toxoplasma include micronemes, rhopteries, and dense granules. As far as Toxoplasma goes, most of you would likely know that it has a very broad host range. So it can infect almost any nucleated cell in its host and can infect nearly every warm-blooded animal. And in those animals, it can cross anatomic barriers. So it can cross into the eye and also can cross into the brain, central nervous system, and crosses the placenta to infect the fetus. However, there's only one known definitive host, and that is the cat. So all felids can be hosts as far as we know, so it includes both domestic and wild felids. So in California, the bobcat and the mountain lion are probably also important hosts for Toxoplasma gondii. There are 3 routes of infection. The first one is via ingestion of sporulated oocysts from the cat. Those contain sporozoites. The second is via ingestion of tissue cysts in undercooked meat. Those contain bradyzoite cysts. Animals that have been implicated as important sources of Toxoplasma infection in humans include pigs and sheep. For the cats, they most likely become infected from birds and small rodents. And the third is via transplacental transmission, and that is the tachyzoite stage crossing into the fetal bloodstream. Regardless of how the infection is initiated, they all undergo this acute phase of infection where the parasite is rapidly amplifying asexually and disseminating throughout the tissues of the host. And then at some point, likely in response to host immune responses, it will actually go into this dormant stage where it can reside for the life of the host. And those are the bradyzoite tissue cysts that form, and they like to form in long-lived cells, so typically in muscle cell or in cells of the nervous system. Within the cat becomes infected by ingesting an infected intermediate host. So birds and rodents, as I mentioned, are the common sources of infection for cats. And once the cat becomes infected, there's this massive asexual amplification that happens within the intestines and the enterocytes. And then some of those parasites that have been amplifying asexually differentiate into the sexual stages. So there's a female macrogamete stage and a male microgamete stage. Fertilization takes place, and that is where the oocyst is formed and is excreted into the feces. Once in the environment, this oocyst is initially shed unsporulated, but goes on to develop, and within 1 to 5 days, roughly, becomes infective. And a single cat, it's important to note, can excrete as many as hundreds of millions of oocysts. So this is pretty remarkable. And as I mentioned, sporulation occurs in the environment, and first there are 2 sporocysts formed, and within each of those, divisions result in 4 sporozoites in each sporocyst. So there are 8 individually infectious entities in a single oocyst. And the oocyst, once in the environment, provides a source of infection for all intermediate hosts. So in the environment, oocysts can accumulate in soil, in water, and in vegetation, where they are a source of infection, as I mentioned, for livestock animals, for humans and companion animals, and also for wildlife. And oocysts are extremely environmentally resistant. So once in the environment, they can survive for months to years, depending on the environmental conditions. So under the appropriate temperature and humidity, they are quite hardy. And in fact, they are very difficult to kill. So treatments that are commonly used for killing pathogens in water, such as chlorination, ozonation, ultraviolet radiation, fail to kill oocysts at concentrations that are routinely used for water treatment. And solutions that are commonly used for cleaning in laboratory settings or in hospital settings, such as quaternary ammonium compounds, ethanol, even 10% formalin, fail to kill oocysts. In the lab, we actually store oocysts in 2% sulfuric acid. So it's pretty impressive just how tough oocysts are. So using a One Health approach, we asked the question, how is a terrestrial pathogen causing such profound disease in a marine mammal? And the goals here were to, 1, develop a model for the transport of oocysts from land to sea, determine how oocysts are encountered by otters in the ocean, to look at what structures or factors are responsible for the remarkable environmental resistance of the oocysts, and then determine if there's a way that we can better identify where oocysts accumulate in the coastal environment to serve as a source of infection to otters. So this first section, I will highlight the work that was done by Liz Van Wormer, and she developed a model to look at the transport of Toxoplasma oocysts from land to sea. To do this, they built a GIS simulation model of parasite loading and parasite transport to look at spatial differences in oocyst runoff, as well as examine how precipitation variability and human land use influenced oocyst transport from land to sea. So they wanted to look at oocyst loading in the coastal environment, and this had to take into account the different feline populations that share that environment. So that would include owned pet cats, outdoor owned cats, wild cats, including bobcats and mountain lions, and then also the unmanaged feral cats. It wasn't surprising that wild felids and feral cats had a higher seroprevalence, meaning more of them had become infected with Toxoplasma, and a higher oocyst shedding rate than owned outdoor cats. But interestingly, because of the sheer numbers of cats, domestic cats had a more significant contribution to the oocyst load in the environment. And using this data, they simulated oocyst-based land-to-sea transmission. And let's see here, I have a graph on the next page. Oh, sorry. And they quantified roughly the oocysts per cell for this model, and that looked at the number of cats times the infection prevalence and the number of oocysts shed. And there were significant differences, as I mentioned, in the infection and shedding across these different groups of cats. And we know that both domestic and wild cats do contribute Toxoplasma oocysts to terrestrial coastal environments. Sorry, I'm having a little difficulty with my advancing. And so this was all plugged into the model, and this area highlighted here is the area that was targeted for the model, and this is the sea otter range, their coastal watersheds. And the next question was, based on what we know from shedding and seroprevalence, what's the estimated number of oocysts that reach the ocean across these different regions within that highlighted area I showed you in that last slide. And so areas where there was high seroprevalence in sea otters for Toxoplasma, that corresponded well with the estimated numbers of oocysts were reaching the ocean in this model. Another question was to factor into this model how coastal development and precipitation influence the pathogen flow from land to sea. And there's a lot going on in this slide, but I will tell you the salient findings were that when you increase precipitation and when there is greater land use modification, so that would be destruction of wetlands, more development, that the oocysts reaching the ocean on this scale here increased quite dramatically. And here is the associated paper if anybody is interested in reading that. So I'll next go to Dr. Karen Shapiro's work, and she was looking at how oocysts are encountered by otters in the ocean. And again, oocysts are shed by these terrestrial hosts. It's just one tiny oocyst. An oocyst is only about 12 microns. And the question is, again, how does it get out into the coastal environment to infect so many sea otters? So Karen's work initially looked at the effect of wetlands and water quality and wetland degradation on this process. So just so you know, wetland water areas provide habitat and food for wildlife. They are instrumental in protecting coastlines from erosion. They can absorb impact from storm surges. And they also serve an important role biologically in filtering out pathogens. This is the associated paper with Karen's work and looking at how wetland degradation affected the flow of Toxoplasma oocysts into the ocean. And she used a surrogate that was developed that had similar size and properties to Toxoplasma that she could actually release into the environment. And there's been very dramatic loss of wetlands in the California area. And not only is this a phenomenon in California, but it is a common global phenomenon. The coastal environments globally are heavily inhabited by humans. And another question that this will lead into ultimately is, how is climate change having an impact in this coastal environment health as well? Karen also looked at, once oocysts do get to the ocean, how is it that they are encountered by sea otters, is there any mechanism for them to concentrate in the ocean? Is there a certain area where they may accumulate? Because if they become just diluted in the ocean, it seems less likely that sea otters would have an opportunity to encounter them. So what she found was that marine snow is an area where organic and inorganic material accumulates. This is something that's been studied by others and in other systems. And she looked to see if Toxoplasma oocysts would also accumulate in marine snow. Snow, because it winds up forming a clump of this material, it gains some weight and it tends to sink and accumulate in certain zones that may help to determine the risk of animals that would be exposed to that. In addition, snow serves as food for invertebrates. And so that's a way in which anything that's accumulating in this marine snow may enter into marine food webs. So this picture on the left is showing, if you're just looking at this picture, you would see that there's a Toxoplasma oocyst there, conveniently autofluorescent in under the fluorescent microscope in the DAPI range. But if you add in here bright field microscopy on top of the fluorescent image, you would see that there's all of this other material present along with the oocyst. And this is a demonstration of how that oocyst has become sort of entrapped in this marine snow. How and where marine snow forms is dependent upon water salinity, currents, particle size of particles that make up the marine snow, but it also has to do with this transparent exopolymer particles, which I will call from here on out TEP. That's an invisible, sticky, gel-like particle, and it serves as the glue matrix of the snow. And this is produced by phytoplankton, cyanobacteria, and this is also produced by kelp. So interestingly, it seems as though kelp produced TEP, and TEP is a sticky kind of glue where Toxoplasma oocysts might become entrapped. So her hypothesis was that T. gondii oocysts will associate with marine snow, and that its association increases as a function of TEP. And this graph here is showing the results of this one study where the proportion of recovered oocysts in marine snow had a linear relationship to the concentration of TEP. So TEP does seem to play an important role. And beyond snow, so there have been a number of studies looking at risk factors for infection in sea otters for Toxoplasma, and one study showed that sea otters who prefer to prey upon turban snails had a higher seroprevalence to Toxoplasma. And sea otters, it's interesting in their behavior, they tend to pick a particular food that they stick with. So they may specialize in crabs or abalone or turban snails. So sea otters that specialize in eating turban snails had a higher seroprevalence to Toxoplasma. And as mentioned, many invertebrate species use marine snow as food, and snails are identified as a risk factor for T. gondii. The odds ratio is 12 times for those sea otters that specialize in snails. And these snails, turns out, are kelp grazers. So Karen looked to see if T. gondii oocyst surrogates could adhere to kelp surfaces, and found that yes, they do. Up to 30% of the surrogates that were released in these experiments did attach to kelp blades that were coated with TEP. And then she also wanted to see if marine snails can serve as a mechanical host for Toxoplasma gondii. So she exposed snails to Toxoplasma, put them in fresh water, and followed up to see if in their tissues there was evidence of Toxoplasma oocyst. And what she found was that they retain oocysts for up to 11 days, and they also concentrate those oocysts to 2 to 3 times the order of magnitude higher than seawater. So they are bioaccumulating and retaining oocysts in their tissues. So it makes sense that sea otters that eat snails would have a greater exposure. Here's a nice picture of a turban snail. So the conclusion from those studies was that Toxoplasma exposure to otters is facilitated by snails because snails can prolong the exposure period, and they also serve as bioconcentrators. So these are the 2 mechanisms that Karen proposed, that Toxoplasma oocysts can concentrate in coastal environments through these 2 mechanisms. One is enrichment in marine snow, and 2 is through the association with kelp on kelp surfaces, and that snails can serve as mechanical hosts. This is another depiction of that story there. Freshwater runoff, oocysts are carried out to the ocean. They associate with macroaggregates, marine snow. They become associated with kelp via TEP, and you have bivalves, turban snails, other invertebrates that can concentrate these oocysts in their tissues. And then animals that eat those, that would include humans who eat bivalves, would be susceptible to infection by this mechanism. The next section of this work that I'll present is the research that I did during my PhD. My questions were, 1, what structures or factors are responsible for the remarkable environmental resistance of the oocyst? My hypothesis was that there are proteins that are present in one or both layers of the oocyst wall that confer environmental resistance. This is a image of Toxoplasma oocysts with a fluorescent microscope. And where do oocysts accumulate in the environment to serve as a source of infection to humans and animals? And my hypothesis was that we could use immunomagnetic separation to concentrate Toxoplasma oocysts in water to better test water and identify sources of oocyst accumulation. This is an image here of oocysts coated in immunomagnetic beads that were developed for this purpose. So first, I just want to tell you a little bit about the oocyst wall. And it is a bilayered structure. So here I'm showing that the oocyst outer wall and inner wall here, so 2 layers. Within it, of course, is the 2 sporocysts. They have their own wall. And then sporozoites are within those sporocysts. If you treat the oocyst with bleach, interestingly, it strips the outer layer of the wall. So that was useful for determining which proteins are present in the outer versus the inner layer of the wall. And when the oocyst is formed within the cat enterocyte, this is a electron microscope image here. There are these wall-forming bodies that are responsible for forming first the inner layer, which is the wall-forming body 2 goes first out to form the outer layer, the contents, and the wall-forming body 1 for the inner layer. So this is probably more detail than you need, but will help to give you the background necessary to understand these experiments. So I took the approach of looking at both gene expression. Conveniently, the entire genome for Toxoplasma has been sequenced and is really well characterized. And so I looked at oocysts that were freshly shed, so day 0, those are unsporulated and immature. Day 4 of sporulation in laboratory conditions, anyhow, about 70% of the oocysts were sporulated, so I call that mid-sporulation. And by 10 days in laboratory conditions of sporulation, they were all fully mature and sporulated. So I looked at using whole genome microarray at the mRNA expression levels across the genome. And I looked not only at oocysts, but I compared that to tachyzoites and bradyzoite cysts from the same strain to see which genes were upregulated in the oocysts at those stages of development. And then I also looked at the protein content of mature oocysts, and I did that looking at those that had been treated with bleach, so the outer layer of the oocyst wall had been stripped, or without bleach, so the outer wall was present. And I also separated the oocyst walls from the internal sporocyst. And I don't think any of this is terribly relevant, but I did identify one class of proteins that were designated tyrosine-rich proteins that were both abundantly expressed in by microarray, but also detected in the oocyst by mass spectrometry, and were not found in the tachyzoite stage. So they were unique to the oocyst stage. Tyrosine-rich proteins are known to form disulfide crosslinks, and in other organisms, under appropriate conditions, form sort of a hardened matrix that protect organisms from environmental stressors. So all of the tyrosine-rich proteins that I identified, they were either abundantly expressed in early stages of oocyst development, sort of mid-stage development or late-stage development, or uniformly expressed throughout. So I wanted to look at those that were expressed early on, meaning that they are likely going into the oocyst wall, and then later on that they may be going into the sporocyst wall. I won't go too much into this detail, but selected a few to look at, and looked at whether or not then I thought they should be in the wall fraction or the sporocyst fraction, and this was supported by proteomic evidence, protein detection by mass spectrometry. So these dityrosine crosslinks, as I mentioned, are known to be present in other organisms, including sea urchin eggs, insect resin, yeast cell walls, and other coccidian oocysts. And they also are thought to be what's responsible for oocyst autofluorescence. My hypothesis was that these dityrosine crosslinks have a lot to do with oocyst environmental resistance. And I developed an antibody to one of these tyrosine proteins and found that they are, in fact, abundant in the wall of the oocyst. So this is immune serum binding to the oocyst. And then they also located to these wall-forming bodies within the macrogamete, and that, remember, is the oocyst stage that is forming in the feline intestine. So I think that this will help us to maybe identify targets for oocyst inactivation, may help us to identify targets for cat immunization to develop vaccines to prevent oocyst development and shedding. So this is very much a work in progress still. The next question that I had was, how can we test water or test other environmental substrates to determine where oocysts are accumulating in the environment so that we have a better idea of where animals and humans are becoming infected? And my hypothesis, again, was that immunomagnetic separation could be used to concentrate Toxoplasma oocysts in water and identify those sources of accumulation. As a bit of a background, the way that we test water for Toxoplasma, I was hoping to model after an environmental, the EPA approved method that's used for Cryptosporidium and Giardia testing. And so the steps there were to develop monoclonal antibodies to the oocyst wall. You couple those antibodies to paramagnetic beads. Those beads are co-incubated with a concentrated water sample that may contain Toxoplasma oocysts, and then you can retain those beads and oocysts on a magnet and wash off everything else. So that's a really good way of concentrating the oocysts. And then you can use direct fluorescence antibody assay for detecting them with a fluorescent microscope. And so this is just an oocyst under DAPI that's autofluorescent. And this is an oocyst that's been labeled with the monoclonal antibody that I developed for this purpose. And then once we've coated the beads with a monoclonal antibody and applied them in this method, here I'm showing that the beads bind well to Toxoplasma oocysts, and that we can, in fact, separate them from the water and concentrate them. So I just want to acknowledge the people that I worked with on these two big projects, and then move into sort of a summary here about what we've learned. And I think that the real take home here is that this study became much more powerful and big and comprehensive because we were able to integrate information across disciplines. We were able to bring people in with so many different expertise and training, and it enabled us to look at studies at an ecosystem level, to look at population health, and then to do laboratory studies. And of course, my studies focused on much more molecular. So this is something that has started at a very small level and then looked at things at a more One Health or ecosystem health level. And as sea otters, they were a convenient animal to focus on because they had such a high infection rate with Toxoplasma. They are also sentinels of environmental contamination, and help us to think about this concept of pathogen pollution. So Toxoplasma is one organism that we've looked at, but we have to consider that likely there are a number of other pathogens that are reaching the coastal environment, and consider how that impacts that ecosystem and human and animal health. And so this is the team that supported the modeling work, and as you can see, it includes engineers, the Bodega Bay Marine Laboratory, hydrologists, marine scientists, and we were lucky to have such a large team to look at this question. And that's all I have today. Is there any questions?
Wonderful. Thank you so much, Dr. Fritz. We will now open the lines for the question and answer session. As a reminder, you may submit questions through the webinar system by selecting the Questions tab on the webinar screen and typing in your question, or you may click on the Raise Your Hand icon on the webinar screen and your line will be Ask a Question. We already have a couple of questions coming in. Dr. Fritz, do you think we can extrapolate this model with Toxoplasma to other pathogens?
I think that's a great question, and I think that making a direct extrapolation, it really depends. Much of what we learned about how Toxoplasma moves into the ocean and how it accumulates has to do with the surface properties of the organism, and as I mentioned, its survival in the environment. So I think we have a lot to learn from Toxoplasma. I think that there will be some differences between Toxoplasma and other types of pathogens, but absolutely, I think it's an indication that there is significant flow of pathogens from land into the ocean. Mm-hmm.
Okay, thank you. Another question. It is my impression that most infections with Toxoplasma are asymptomatic. Do the other otters become ill, or is the negative impact upon this only related to fetal deaths?
Oh, so for sea otters, the extent to which it causes fetal death, or the extent of transplacental transmission, I think we're still learning about that. So Toxoplasma is actually causing significant encephalitis and clinical signs in adult, otherwise healthy sea otters. And what makes one sea otter go on to develop clinical disease versus another that remains healthy with a Toxoplasma infection, I think we don't entirely know. But yes, the infection and disease is very significant in adult sea otters.
Okay. You mentioned that some sea otters preferentially feed on snails. Are these different diets found at the species or subspecies level, or do individual sea otters truly have different food preferences?
Yeah, that's interesting. It does appear as though individual sea otters have their individual food preferences. It is probably a learned thing that was how they were taught to feed, and that may help them to within their community to develop their own niche. So yeah, that is within a community of sea otters that some have different prey. They have different prey preferences.
Okay, thank you. Next question. Are there good candidates for targeting those dityrosine bonds for oocyst inactivation, and is there any data indicating efficacy?
Right, that is something that I've taken to chemists and molecular biologists and other people to ask if they have any idea what we can do to inactivate those bonds. And at this point, it's not known. We don't have a good target yet. We are looking at what happens if you do disrupt the tyrosine. So looking at wiping out the gene that is responsible for that tyrosine protein to see if that impacts whether or not the oocyst wall can form and whether or not the oocysts still have their resistance to treatment with bleach and chlorine and such. So that is still an open question.
Is there any human risk of getting toxoplasmosis from consuming cooked or uncooked bivalves?
Certainly from ingesting uncooked bivalves, yes, I would say that there is a risk. It has been shown by many that bivalves can concentrate Toxoplasma oocysts in their tissues. In humans, most infections are mild or asymptomatic unless there is some immunocompromise. So immunosuppressed individuals more often will have clinical infection. But yes, the risk of becoming infected by eating undercooked or raw bivalves is there for people.
Okay, next question. Has there been a serologic evaluation of T. gondii infection in other marine animals or species, mammalians or fish and crabs in the sea otter environment?
Yes, so there have been serologic tests of other marine mammals, and there is evidence of infection in almost every other marine mammal that's been tested. Sea otters may be at greatest risk because they are in more near-shore environments, but other marine mammals, including sea lions and harbor seals, and quite a long list. But yes, there's evidence of infection serologically in those animals as well.
Okay, thank you. This is a two-part question. What was the prevalence of oocyst excretion in wildcats, and can subspecies strain varieties of oocysts be used to determine the source in the environment?
Okay, I don't remember off the top of my head what the seroprevalence was in the wild felids, but there has been quite a lot of work done to look at genotypically characterizing isolates from that are recovered from felids and also from sea otters, as well as those that are recovered from marine bivalves or from other environmental matrices to try to tease out patterns of transmission. And so that is done at the genetic level, and there are several papers available, and I'd be happy to, if somebody sends me a message, to share any of those papers.
Wonderful, thank you. I think we have time for one more question. Dr. Fritz, what risks do you think toxoplasma in the ocean poses to people who spend time in the ocean recreationally?
I do think that is also a great question, and one that hasn't specifically been looked at, but presumably based on the exposure that we know, sea otters are at risk to other mammals that use that same environment would likely have similar exposure, but that hasn't specifically been looked at. I know people have been interested to see if maybe people who tend to do a lot of snorkeling or surfing or engage in routine activities in coastal environments have a higher seroprevalence.
Wonderful. Thank you so much, Dr. Fritz. On behalf of CDC's Clinician Outreach and Communication Activity and the American College of Veterinary Preventive Medicine, I would like to thank everyone for joining us today with a special thank you to our presenter, Dr. Fritz. Free continuing education is available for this call. If you would like to receive continuing education, you should complete the online evaluation by March 8th, 2018 using course code WC2922. For those who will view the archived webinar after March 8th, 2018, please complete the online evaluation between March 9th, 2018 and March 9th, 2020 using course code WD2922. All continuing education and contact hours for COCA conference calls are issued online through TCE Online, CDC's training and continuing education system at www.cdc.gov/TCE online. To receive information on upcoming COCA calls, subscribe to COCA by sending an email to coca@cdc.gov and write subscribe in the subject line. Also, CDC has launched a Facebook page for clinicians. Please like our page at facebook.com/CDC Clinician Outreach and Communication Activity to receive COCA updates. If you have any questions, please email us at coca@cdc.gov. Thank you again for being part of today's webinar. We hope everyone has a nice day.